All right. So we'll wait for others to join. Good afternoon, Nehal.
Good afternoon, sir.
All right. Let me share my screen and then we will start. All right. Screen share. Okay. This is where we let off in the last class, right? We talked about how we express proteins in E. coli, and the last system we talked was about this maltose binding protein, and how do we eventually purify the protein. Purifying the protein is a fairly big topic. Okay, so we will probably towards the end of the this thing we'll spend some time on how proteins get purified, right? Obviously, the insulin that gets injected into patients or any of those antibodies, that get fused, put into patients, they need to be highly pure, highly pure. Okay. So after expressing, purification is as much a science as much it is an art. So it's a combination of. I mean, I don't. When I say art, it doesn't mean that people start looking at art from that artistic sense. It is not that. you know Art actually involves using your knowledge, and different people can do purify the same protein in different formats, and end up with having the same purity, same applications. And so, as much as protein purification is a science, it's equally an art. How do you choose different kinds of matrices for separating your protein from rest of the cellular protein and all that? Okay, so we will spend some time, hopefully, towards the end of this module or in module 3. Okay, so till the previous class, we looked at how you can express proteins in E. coli, right? We typically use PET system. PET system is just one of the systems, but then you have so many different companies that have their own expression systems, and people have been extremely successful in expressing different kinds of proteins in bacteria. Okay. So is there any knowledge ki whether my when I want to express my protein, whether it will work in bacteria or yeast or in mammalian cells? At this point in time, the only thumb rule is that if your protein has extensive post-translational modification, which typically would be glycosylation, okay? If it is highly glycosylated protein and glycosylation is essential for the function of the protein, then by and large, E. coli system will not work. Okay. But there are many proteins, eukaryotic proteins, that are glycosylated, yet when you express it in E. coli, it works. Okay. So it is not always this thing. Okay. So you still need to try and express things and see whether it will work or not. But as I said, if your glycosylation and your protein function are linked together, then you can be pretty much sure that expressing it in E. coli will not make any sense, except for maybe raising antibodies. Okay. So that being the case, the next system that people generally try to use is express your foreign genes in yeast. Okay. Now there are several choices when we decide to express our protein in yeast expression system. Saccharomyces cerevisiae, we all know that is something that is so common, right? Baker's yeast, right? You also have another species called Saccharomyces pombe, which is very close to Saccharomyces cerevisiae. So, many of the tools that are used for Saccharomyces works very well with Saccharomyces also. Okay. Then you have things like Trichoderma ricci, which is very well known. Any idea why Trichoderma ricci is so well known? Or I hope you have heard this term before. Any idea? Does this fungus, the name of this fungus rings a bell?
Sir, it's also known as brewer's yeast.
Not really, not really. So Trichoderma ricci is, who was this? I am not able to see your this thing.
Oh,
Who
I'm
was this?
sorry. I thought you were asking about the Saccharomyces
No,
cerevisiae.
no, I was asking about Trichoderma ricci. Okay. So Trichoderma ricci is a fungus that is extremely, good in growing in rotting trees. Okay. So it has extremely high cellulose activity, right? So it essentially breaks down the cellulose, which is the most common biopolymer in the entire planet. But it is also one of those biopolymers that are not easily degradable. But Trichoderma ricci is something that routinely grows and it has very high cellulase content. Okay, then you have another fungus called Pichia pastoris. Okay, which is extremely handy in under certain situation, and in fact it has now replaced Saccharomyces cerevisiae as one of those common tools for using expression, okay, protein expression. Then you have another thing called Hansenella polymorpha, and then you have Aspergillus nidulans, which I'm sure all of you must be aware right from your, high school days. Okay. Aspergillus nidulans is an organism that makes lots of vitamin C. Okay. So in fact, many people use the commercially produced ones are the chemically manufactured ones are essentially derived from Aspergillus nidulans. Okay. Now, irrespective of whether you choose Saccharomyces, Saccharomyces, or any of these other organisms, most genetic manipulations are done in E. coli before transforming the yeast of your choice. Okay, so essentially you will be using shuttle vectors because cloning and doing all these manipulations is much easier in E. coli than doing it in yeast. Okay, so you essentially will be using a shuttle vector, doing all the manipulations in E. coli and then transforming that into the yeast. Okay. The choice of yeast, whether you want to use Saccharomyces or Saccharomyces or any of the other things, to a large extent depends on how much expression do you want, okay, and what will be the substrate that you'll be using for induction. For example, when we talk about Saccharomyces or Saccharomyces, you could use a variety of substrates for inducing your gene of expression interest. Okay, when you think about Pichia pastoris, it is typically which uses methanol. Something that, remember, we used to use IPTG for expression in E. coli. Similarly, for Pichia pastoris, you will be using methanol. Okay, you will be growing essentially the fungus in methanol. Okay, you will grow it under certain conditions, and then you will be literally be forcing the fungus to use methanol as its sole carbon source. No glucose, no lactose, no other sugar, but essentially it breaks down the methanol as its sole carbon source. Okay. So it depends. It depends on, what is the kind of expression. Some proteins are stable, some proteins are not very stable under those conditions. So you may still need to play around and find out which one works well for you. Okay. Now there are certain advantages and disadvantages. Saccharomyces cerevisiae, as we all know, it is unicellular. It's an extremely safe organism. It's readily amenable to genetic manipulation. Okay, it has a minimal level of post-translational modification. Okay, so it does do glycosylation, right? This glycosylation comes under variety of formats. Okay, so the way yeast glycosylates protein versus the way humans glycosylate the protein are very different. But to a large extent, it is substitutable. Okay, so So to a large extent, but not always, to a large extent. Okay. And one of the beauty of yeast is that it actually secretes. You have this ability to, secrete the protein into the growth medium. So you don't even have to, which makes purification very easy, because once the major component of these unwanted proteins is from the cells, and once the protein gets expressed and it is into the growth medium, all you need to do is give a low speed spin, all the yeast will come and form a pellet, take your supernatant, and you can very easily purify the protein. So protein purification process becomes easier. Okay. But it also comes its own disadvantage. The expression levels are not that very high. Okay. And invariably, many a times you have hyperglycosylation. Many a times, the proteins have multiple, glycosylated these things, and the protein may or may not, have the same characteristic that is essential for its function. For example, suppose let us say you've expressed insulin, recombinant human insulin yeast, and it is showing a different glycosylation pattern. We don't know whether it will be functional in the humans or not, or it may have some unusual adverse effects. It is possible that those glycosylation patterns may generate some adverse effects. Okay, so we have to be extremely careful. More importantly, to a large extent, the plasmids are unstable. So, but this can be overcome. This can be overcome because people now have, plasmids that integrates into the host genome instead of, putting it as a plasmid, which is typically the case with E. coli. Okay. In yeast, you can make sure that this plasmid gets integrated into the genome, and then it can have constitutive expression. Okay. Now, the methylotropic yeast, so things like Pichia pastoris, and I will tell you a few more names, the ones that use methanol as their sole carbon source instead of glucose or instead of any of those other things, these are called as methylotropic yeast. Okay. They have a strong and a regulatable promoter. That is their advantage. Okay. The moment you add methanol, certain promoters get expressed at a very high level. Genes under certain promoters get expressed at a very high level. Okay, so if you can genetically alter and put your gene of interest under those promoters, they're going to be expressed at a very high level. Okay, so you have a very high level of expression, and it generally doesn't have a protein secretion. But what people have done is to use this mating factor from Saccharomyces cerevisiae, okay, and use that and make sure that the protein gets secreted. So you essentially will be having the mating factor sequences, which is the one that is responsible for putting out the protein into the extracellular medium. You'll be fusing that along with your gene of interest. So when it gets expressed, it will come with this pre-pro sequence, which then gets cleaved at the interface of the plasma membrane, and then your protein gets expelled into the medium. Sometimes it will come along with the pre-pro sequence, which you can cleave later. Okay, that is not a big issue. Okay. More importantly, these methylotropic yeast have integrative plasmids, which means when you clone these gene of interest and put it in the plasmid, it will invariably go and integrate into the host genome. Okay, and then you have post-translational modification, which are N-linked and O-linked glycosylation. I'm sure you would have read in your class on biochemistry class what are the post-translational modification, what is N-linked and what is O-linked glycosylation. If not, I can spend some time, not a big deal. Okay, and generally doesn't have any hyperglycosylation. Okay, so these are some of the reasons why people have switched from Saccharomyces cerevisiae into methylotropic yeast. Okay. So one typical example of methylotropic yeast is Pichia pastoris. Okay. So this has become, one of the favorite tools, not only in academic labs, but also in the industry. So people have started using Pichia pastoris as a expression system. And so this organism uses methanol as its sole carbon source. Typically, every organism, uses sugars, the carbohydrates, right? Lactose, glucose, maltose, or any of those things as its sole carbon source. You can have organisms that can have proteins, only proteins as the sole carbon source. All of you, I'm sure, must be aware of different diets that people consume, humans consume. There are diets that are rich only in proteins, okay? No fat, no sugar, nothing, only protein, 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 high protein diet. Different diets are there, right? All of you are aware. And like that, for Pichia pastoris, the sole carbon source or the sole energy source is going to be methanol. Okay. The moment you put methanol, so this picture, is my cursor moving? Can you see that?
Yes, sir.
Okay. So this methanol gets metabolized in a structure called peroxisomes, okay, which is found inside these methylotropic yeast, okay. The moment methanol enters, alcohol oxidase, okay, alcohol oxidase gets induced and converts methanol into formaldehyde, okay. Now in the process, it also generates a lot of hydrogen peroxide, which is converted back to water and oxygen by an enzyme called catalase. Okay, so otherwise, if you look at it, hydrogen peroxide is a highly toxic molecule. It brings a lot of oxidative stress to the organism. But because of the expression of catalase, okay, this oxidative stress is taken care by the organism. This formaldehyde now can become formic acid or formate by the action of formaldehyde dehydrogenase. And then this formic acid or formate gets converted to CO2 by formate dehydrogenase. Or this is a minor pathway. Majority of this formaldehyde is acted upon by xylulose 5-phosphate, which converts it to dihydroxyacetone, this thing, okay, which is taken care, and this goes into your TCA cycle, okay, dihydroxyacetone phosphate, okay, so it gets into the TCA cycle, and that is how methanol acts as a sole carbon source, okay. As I said, one of the first thing that happens is the alcohol oxidase promoter gets activated. Okay, the alcohol oxidase enzyme is literally 30% of the protein in the organism is alcohol oxidase. Okay, so the alcohol oxidase promoter must be extremely strong promoter for it to be expressing such high levels of alcohol oxidase. So people have taken advantage of that. Okay, and now you can clone your gene of interest downstream of alcohol oxidase promoter, and you can induce in the presence of methanol. In the absence of methanol, this doesn't get induced. Only in the presence of methanol, this gene gets induced. Okay. And as I said, your protein can be literally 35% of the fungal protein, cellular protein can be your protein of choice. Okay, and the growth medium is generally non-conducive to other contaminants. Typically when you use Saccharomyces or any of those other yeast, the pH is held close to 7, which is also something that is conducive for bacterial contamination, which is also conducive for other contaminants. But in this case, the pH is kind of acidic. Okay, it's anywhere between 4 to 6, and because it has a lot of methanol, many of the organisms don't grow except for this methylotropic yeast. And as I said, you can have extremely high levels of protein expression. Okay. So these are the examples of methylotropic yeast: Pichia pastoris, Pichia methanolica, Pichia angusta, Candida boydini. Okay, these are some examples of methylotropic yeast. Okay, and very briefly, 1969, a scientist named Koichi Ogata found Pichia pastoris. Okay, and he studied it briefly before, this company called Phillips Petroleum Company realized the value of this Pichia pastoris. Just imagine a petroleum company, having interest in yeast. Okay, because they realized that this yeast could be a very valuable system. Okay, and this turned out to be an attractive source of single cell protein. So you can actually grow this methylotropic yeast Pichia pastoris in huge fermenters. You can dry them. Okay, and you can give it out as a source of protein supplement. For people who suffer from protein deficiency in certain regions of the world. Okay, this can actually be a good source of protein for them. The once they consume this Pichia pastoris capsules, it comes in the form of a capsule. It can also go in the form of a syrup. Okay, the cells get lysed inside your stomach, and the proteins get digested into amino acids. So this can actually serve as a source of single cell protein. Okay, and there are a lot of different kinds of media available, and protocols have been tweaked so that you can literally grow this at a very high density, extremely high density. So this high density is needed when you want to make this out not only for your protein expression, but also when you want to make other applications, like if this is going to be used as a protein capsule. Okay, and heterologous protein expression is under the influence of alcohol oxidase, as we saw, and many genetic manipulation protocols have been developed. Several strains have been developed. Okay, there are strains which are called as MUT plus or methanol utilization plus. Okay, and they have increased levels of this alcohol oxidase promoter, much better than your wild type. Okay, and then you have another set of strains called MUT S. The S is a superscript. It is essentially it's deficient in a protease. Okay, when you try to express many of these proteins in whether E. coli or any expression system, the proteases that are found in every cellular organism invariably goes and starts degrading your protein of interest. Okay, so you will end up losing some amount of protein for these proteases, and this strain of Candida or Pichia, some of these strains are deficient in these proteases. Okay, and then you have a variety of vectors. As you can see here, that are available for cloning, and you have both very strong promoters which can be either inducible or it could be constitutive. See, you have the choice whether you want to express this protein all the time, or whether you want to let the yeast grow to a very high density and then switch over the carbon source into methanol, right? You can grow these yeast under normal growth condition with glucose or with any of those highly nutritious this thing carbon source, and then once it has grown to a very high level, you could replace that carbon source with methanol, right? So you can actually see that you will be able to get much higher yield of your protein. Okay. Any doubts as of now? Can we move ahead?
Sir, can you repeat the inducible and constitutive part again?
So when you try to express your gene of interest, you have a choice whether you want to express it constitutively. Constitutively means it is going to be expressed all the time. Just imagine your normal cell, which is continuously it is expressing certain genes at a constitutive level, right? So you can have your gene of interest under constitutive promoter. That means at every growth stage, your protein is going to be expressed. Okay, many a times This could be, advantages, but there are certain times when it could be disadvantages. Okay. So you have the choice whether you want to put it under a constitutive promoter or under inducible promoter. For example, the alcohol oxidase, AOX. Okay, this is an inducible promoter and it gets induced with methanol. Okay, whereas GAP is glyceraldehyde 3-phosphate dehydrogenase, which is involved in your TCA cycle. This is going to be expressed constitutively, always getting expressed. Okay, I forgot that this thing for FLD. I will come back and check and I will come back to you. If not, please remind me. Okay. So these are the examples of inducible and constitutive promoters. For example, you could you could clone your gene of interest under inducible promoter. You can grow them to very high densities like what is written here, now 130 gram per liter, and then you can induce it. When you do that, you're going to have extremely high levels of your recombinant protein being produced. Okay? Does that answer your question? Was it Shakti?
Yes, sir.
Okay. All right. So this is a system that is commercially available from a company called Invitrogen. Okay. So typically, they have this different kinds of vectors. Okay. And this is, as you can see, it is being driven by the alcohol oxidase promoter. Okay. You will be cloning your gene of interest downstream of this promoter. Okay. And the selection is by an antibiotic called zeosine. Okay. Now, you would clone your gene of interest, you could transform it, you will be plating the transformants on medium containing zeosine, and you will select about 10 to 20 colonies, and whatever is growing on zeosine containing this thing, and then you will check whether these clones are expressing your protein or not. Okay. And then once you know that this is the clone that is expressing my protein at a very high level, you can start growing them up in higher quantities, right? So this actually gives you an example of two different promoters. One, the AOX promoter, which is an inducible promoter, which gets induced only in the presence of methanol, and then you have GAP promoter, which is glyceraldehyde 3-phosphate dehydrogenase promoter, which is a constitutive promoter. Okay, so you could clone. So in this case, you can see it is they have cloned the alpha factor upstream. So this is your multiple cloning site where you will be cloning your protein. This is the alpha factor, the signal peptide. So it gets secreted into the medium. It is not going to be retained in the cell. Okay, you have two different choices. One in which you have this and one in which you don't have this. So you can decide, how do you want to do this? Okay. Similarly is the case for the constitutive promoters. You can decide whether you want to do it this way or that way. Okay. Okay. So that brings us to the end of yeast protein expression system. We have discussed very briefly about Saccharomyces cerevisiae, and then we discussed mostly on Pichia pastoris because that's something that is being used commonly. Okay. Many a times, even after doing all this, your protein may not be functional in yeast. Okay, when that is the case, then you would start expressing it in what is called as a baculoviral expression system. So what is a baculovirus? A baculovirus is something that infects insect cells. Okay, just as you have mammalian cells, you also have insect cells. Okay, and it is an ideal vector for genetic engineering because these viruses are non-pathogenic to humans. Okay, and they have certain strong promoters which codes for proteins that are not essential for the viral production. Okay, that means under these promoters you could put your gene of interest and the virus will behave normally. Okay, and you can get up to 40% of the total protein as your target protein. See, we talked about 35% in Pichia pastoris. Okay, here we are looking at 40%. Okay, so this is the typical life cycle of a baculovirus. Okay, so these are called as enveloped viruses because when they come out of the host cell, they carry a piece of the host cellular membrane. These are called as enveloped viruses. Okay, when these viruses, encounter an insect cell, okay, SF9 is one particular insect cell. When it encounters, it fuses. This one essentially facilitates in the fusion, okay, and enters into the cell, okay, and once it is internalized, it results in uncoating, and then the genetic material gets into the nucleus of the cell, okay. Inside the nucleus, okay, there is, you have transcription and translation, and then, you know, the proteins are targeted towards Golgi, okay, and then you have these things, you know, virus replicating, and then it tries to go out with your cellular membrane. So that is how you have what are called as budded viruses or enveloped viruses. Okay, and these are the receptors that it carries. You know, when it comes out of the insect cell, you know, it is carrying different kinds of receptors from the membrane. Okay, so it becomes easy for it to fuse to the insect cell again. So there are three different insect cell lines that are very commonly used in the laboratory. SF9 is the most commonly used one. SF21 is also commonly used, and then you have a few laboratories and certain commercial institutions that use what is called as Hi5 cells. Okay, the difference between SF9, SF21 and Hi5 is the doubling time, right? SF9 takes almost 72 hours for it to double. SF21 takes about 24 hours, and Hi5 cells take about 18 hours. Okay. So the foreign genes are cloned under the control of either of the two promoters. That is, there are two very strong promoters. One is called as a P10, and then one is called as the polyhedrin promoter. Okay. So you could clone it under either of them. You could clone it under Pol10, either polyhedrin, or under P10 promoter, and you will have very high levels of expression. Okay. The patent on the baculoviral expression system has expired. So a lot of commercial applications have started emerging for this insect cell line expression system. And I just thought that I should show you this, you know. So these are products that have been approved for commercial use, and these typically use the baculoviral expression system, including a vaccine called Flublok, okay, which is against the flu virus, and this was approved in 2013, okay. And similarly, you have many other things that are, expressed in baculoviral expression system, and it is being used in humans and in other animals like pigs. Okay. One notable thing is this vaccine called Cervarix, which is against papilloma viruses, right? And this has caught this imagination of many of the young adults in the European continent, in the US, and also I think this is probably approved in India also. Okay, so how does this happen? So you basically this again, baculoviral expression system is sold by Invitrogen and several other companies. Okay, each one has their own way of doing things, but essentially, this is your gene of interest, whatever clone you're trying to express. This is cloned under the certain clone called entry clone, and it is cloned between two regions called AT1 and AT2. Okay, AT-L1 and AT-L2. Okay, and then you have specialized E. coli strains that carry this DNA. Okay, that carry this DNA in them. Okay, so you basically transform this in the E. coli strain, and within E. coli you have recombination happening at the AT locus. Okay, and then typically what happens is your gene of interest gets into the this thing. Okay, and then this can be introduced into the insect cell. You need to introduce into insect cells. See, with E. coli or with yeast, you do what is called as transformation. Okay, the process of transferring an exogenous DNA into E. coli or yeast is called as transformation. The process of introducing foreign DNA, okay, into mammalian cells, has a different name. It is not called as transformation. Instead, it is called as transduction. Okay. So you have to be careful. You can't say I have transformed mammalian cell. Okay. A transformed mammalian cell becomes cancerous. Okay. So that is why people don't use the word transformation. Instead, they have transduced. Okay. Did you all get the difference between transformation and transduction?
Yes, sir.
So once you transduce these foreign gene into these insect cells, okay, you grow them. Typically, the transduction efficiency is very low, you know. So only few cells get infected in the first time, okay, and then you would, and the viruses are in the medium, right? So you basically collect the first viral stock. So P1 stands for the first viral stock. Then you take this virus and then infect a fresh batch of cells. So typically what happens is the viral number increases, you know, the viral number increases. And then from the second viral stock, you know, you go to a large, this thing, you know, you now start growing them in big fermenters or big flasks, okay. And then, you know, you infect these insect cells again, okay. And you will generate what is called as a third viral stock. Okay. Now, third viral stock is something that is extremely enriched, and these viral particles are essentially carrying your gene of interest. Okay. Now, when you infect from the third viral stock, when you infect these insect cells, okay, you infect them typically at what is called as multiplicity of infection. That means for each single insect cell, you're trying to infect each single insect cell with a 5 to 10 different, 5 to 10 times the amount of virus. So you will essentially be calculating how many insect cells you are having in your medium, and then accordingly you will add 5 to 10 times more viruses. Okay, you can there are certain techniques by which you can count how many viruses are there. Okay, so after enumerating, you will be infecting the insect cells with 5 to 10 times, and then anything, you know, the cells that is expressing your foreign protein, you can actually see they start making some vacuolar appearance. So that actually tells you that your foreign genes are getting expressed. Okay, so then you would lyse the cells, you could harvest the cell lysate, and you can proceed to protein purification. So this is typically the workflow with baculovirus. Okay. What are the advantages? The polyhedrin gene is not required for the continuous production of infectious viruses in the insect culture. So it can be replaced with that of your gene of interest, your heterologous gene. Okay. This polyhedrin gene is a promoter is extremely strong, and so that results in extremely high level of recombinant protein being produced. Okay. And this system of baculovirus also does post-translational modification, which is better than your yeast, which is better than your Pichia pastoris. Okay. As usual, it will have its own disadvantage. One is that it's kind of expensive, you know, it's expensive, and the glycosylation pattern is again different from the mammalian systems. Okay. And sometimes, you know, the baculoviral infection can start killing the cells very quickly. And so each time you will need to first grow the insect cell and then infect them with the baculovirus. So that is the case with baculovirus, right? And it is not really a this thing for commercial production, but people have overcome this with specialized cells, and they have been able to make it commercially feasible. By and large, you know, it has this disadvantage. Okay. Now we will move into mammalian cells. So there are several ways by which a foreign gene can be introduced into mammalian cells. I put animal cells. Typically, you know, when we talk about mammalian cells, people use few cell lines. Some of them are of mammalian origin. Some of them are from different other animals. Okay. Now, the way by which you can introduce a foreign gene into the mammalian cell, can be broadly divided in two categories. One is called as instrument based and the other is called as reagent based. So that means there are chemical ways of introducing a foreign gene, or there are physical ways of introducing a foreign gene. Okay. Now instrument based will come under physical. For example, you could electroporate, we are going to talk about electroporation later, so I'm not going to put emphasis on electroporation. You can have gene guns, you can have micro injection, you have many different kinds of ways by which you can introduce a foreign gene into a mammalian cell. Chemical based, for example, you can have certain chemicals like calcium phosphate, you can have certain lipids, okay? You can have things like DEA, dextran. Now, all these things have a positive charge. Calcium phosphate, DEA, dextran, these lipids are extremely positively charged and your DNA is negatively charged. So invariably, they form a complex. Okay. When this complex, of this positive and negatively charged molecules is introduced into an inside a cell. Inside a cell, they dissociate. Okay, and the DNA gets taken up into the nucleus and it can get expressed. Okay, the third way by which we can do this is biological. For example, viral based. Okay, all of you are aware of the corona vaccine, right? How is that done? It basically you give grow your cells, okay, and these viruses will infect your cells, okay, and they will multiply, and then your immune system sees it, and then it mounts an immune response. So what these viruses have, they have the ability to take the foreign gene into any cell type and it can express. Initially, people used many different kinds of vectors. Okay, viruses as vectors. We will see a short list towards the end. Okay, so transfection, is a process by which foreign DNA is introduced to the cells, and this itself can be classified either as transient or as a stable transfection. Okay, in transient, genes get expressed only for a short time, anywhere between 24 to 72 hours. Sometimes you can stretch it up to a couple of days. Okay, and the transgene expression is very high. And typically, remember we talked about zeosin selection, we talked about ampicillin selection, we talked about neomycin selection. There is no need for selection. Okay, and the plasmid, it is an episomal expression. When we talk about stable expression, genes continue to express beyond 72 hours. And initially it is very high to begin with, but then, as time goes by, you will have, low level of expression. Okay. And typically you will be using certain selection markers here. And it is, the low level expression is because a part of the plasmid got integrated into the genome. Okay. So this is typical example. When you do transient expression, cells are expressing the green fluorescent protein. That's why you are seeing every cell, lighting up green. Okay. Within the first 24 to 72 hours, up to 3 days, okay, you will see that, the expression is very high, very high. This can actually be stretched up to a week, okay, up to a week, not beyond that, okay. Now, this is something where these cells, when you leave them for a longer time, okay, these very cells, when you leave it for a longer time, most of the cells stop expressing GFP, but some of them will still continue to express GFP. And when you leave this for even longer time, those cells which have integrated this plasmid, they will be expressing this protein at a fairly high level. Okay. So these are some of the vectors that are used in mammalian cell transfection. Okay. So when we talked about transformation, we talked about transduction. Now we are talking it as transfection. So transformation is for bacteria and yeast. Transduction is typically used for baculovirus, and transfection is used for mammalian cells. All right. Did I make it clear?
Yes, sir.
Yes, sir.
Okay. So this is the structure of expression vector for mammalian cell. You know, it carries kanamycin, neomycin resistance. It carries a strong cytomegalovirus promoter, which is expressed only in eukaryotes. Okay, so you would clone your gene of interest here. There is a poly A signal. Okay, so at the end of your transcription, when it gets translated, the ribosomes will know that this is the end of the gene. Okay, this is one vector. Right? This is something that I had shown you earlier when we talked about shuttle vectors. This is PIRES vector, which carries two MCS multiple cloning site. Okay, and you could clone your gene in either of them. This again is cytomegalovirus promoter based one. Okay, it's a retroviral vector. Okay, this is something that is unusual. Okay, people have started using this, and then these are another class of vectors. Okay, which carry viral elements. Okay, for example, the 5' long terminal repeat, the 3' long terminal repeat, and then you have a short, again this also uses CMV promoter, and then you have a short multiple cloning site where your genes get expressed. Okay. Biological methods, these are the viral vectors that are used. For example, people have tried to express the gene of interest in murine leukemic viruses. This is still in the experimental stage. Okay, people have tried to modify the HIV virus and try to use it for expressing the gene of interest. They have done HTLV. So these are RNA-based viruses, right? You can also have DNA-based viruses. For example, adenovirus, adeno-associated virus, herpes simplex virus. All these viruses can be manipulated to have your gene of interest, and they will go and infect only cells, okay, select cells, and you can have expression of your recombinant proteins. Okay. Adenovirus, these are viruses which carry double-stranded DNA. So you could, in theory, clone your gene of interest under certain promoters, and you could express it in literally every cell type. Okay. Adeno-associated viruses are single-stranded DNA viruses that specifically insert their DNA into chromosome 19. Okay, chromosome 19 is the most commonly, this thing plays where the genes get integrated. Okay, so this could be useful for people who are, who have genetic deficiencies, who have, genetic diseases, right? So you could try to do gene therapy with these vectors. Okay. HSV is herpes simplex virus. This is again a double-stranded virus, but it has affinity for neurons. Okay, it doesn't infect every cell type. It infects specifically neurons. Okay. Okay. So this basically gives you the properties of different viral vectors. In the case of retroviral vector, your insert size cannot be greater than 8 kilobase pairs. Okay, and it essentially results in random insertion. You have a series of vectors called lentiviral vectors, which can infect both dividing and non-dividing cells. Okay. Typically hematopoietic stem cells, cells from the blood lineage, right? They're all dividing cells. Cells from certain origins are dividing cells. On the other hand, terminally differentiated cells are called as non-dividing cells. Okay. This can carry up to 9 kilobase pairs. Adenovirus again has up to 8 kilobase pairs. Adeno-associated virus can carry around 5 kilobase pairs, nothing more than that. Herpes simplex virus can carry up to 40 kilobase pairs. Okay. And vaccinia virus can carry up to 25 kilobase pairs. Okay. Some of them have transient expression, some of them have stable expression, some of them will go into very specific location. For example, AAV, adeno-associated virus, can go into the chromosome 19, okay, and get expressed. Okay, so this is how you end up doing it. So this is, you have a expression vector where you have cloned your gene of interest, okay, and you will be transfecting the this DNA to mammalian cell, right? In this case, it says A293. This is called as human embryonic kidney cell, HEK293. Okay, you will transfect, and then about 48 to 72 hours after this, you will be collecting the supernatant. Okay, and then you can determine the viral stock. Okay, whether the virus has multiplied, and if it not, how much, if it has multiplied, how much it has multiplied. If not, you will have to restart the entire process. Okay, if the virus has multiplied, then you could collect the virus and you can redo the entire process again till you have enough cells that are infected. Okay. Electroporation and other things we will probably look in the next module when we talk about introducing foreign genes into cells. Let me see if I can come up with some lecture for how to purify protein. We will do this tomorrow. Okay, so any questions on today's class?
No, sir.
All right. So if there are no classes, I'm still compiling. So maybe either tomorrow or sometime I will mail it to you. Okay. All right. So if there are no further questions, then I would stop my class here, and then we'll see tomorrow then. Okay.
Thank you, sir.
Yeah.
Thank you, sir.
Vanshika, are you there? Devanshi, just check whether Vanshika is there or not, and then let me know. Okay.
Sir, actually, Sunny sir told me she is facing some problem in attending the classes. I don't know what happened to her.
ठीक है. Okay, fine. I'll check with Dr. Sunny then. Okay.
Okay, sir.
Thank you.
Thank you.